Perfect.
Good. So as Victorio said, my presentation is going to be on ketones and the way we've used them in mild cognitive impairment and the relationship that we see between the cognitive improvement and various forms of multimodal imaging that help dissect some of the mechanisms that are involved. These are my disclosures. So when we think about dietary interventions, some of you have been discussing, I'm sure, yesterday and later today, the interventions that are classically now known in the area of cognition and brain aging, which is to say the finger study, which was a multimodal, of course, intervention, the Mediterranean diet, and the mind, which is a form of Mediterranean diet intervention. And these approaches have a few points in common. We're trying to raise B vitamins, which are important for the brain. We're trying to increase omega-3 fatty acids, and probably in most cases, whether it's intentional or not, but in most cases, it's to reduce the pressure on glucose metabolism, to reduce the dietary carbohydrate. And what I want to add to this mixture this morning is that a ketone strategy is definitely, in my view, important to helping meet the brain's energy needs, which are pivotal to optimizing cognitive health during aging. So it's a combination of improving structural elements within the brain, but also meeting the deteriorating brain energy condition. And this we're taking the approach with ketones to achieve that. So when we think about cognitive issues during aging, obviously Alzheimer's disease comes to mind. And in the area of the conditions that contribute to developing Alzheimer's disease, glucose metabolism in the brain is it's a significant, and it's a well-known problem. This has been known for over 40 years now. So these are PET scans of fluorodeoxyglucose uptake, so glucose metabolism in the brain, and they can be used to calculate that the brain is consuming about 100 to 125 grams of glucose per day. In Alzheimer's disease, these cross-sectional scans show the parietal lobe with the arrows where there's a definite decrease in glucose uptake. At the early stage of Alzheimer's disease, the first objective stage of Alzheimer's is called mild cognitive impairment. And this problem identified by the arrows here, the lower color, the greenish color as opposed to the yellow to orange, indicates a problem with glucose uptake on the order of 10%. So about 10 to 12 grams of glucose per day is not getting into the brain. And in Alzheimer's disease, this progresses to above 20%, and when the disease is moderately advanced, can be on the order of 40 and even 50%. So this is a major problem with brain function. And it has been generally considered to be a consequence of the disease. And what I'll be showing you today is that this, in fact, is contributing to it. So our view of this contribution of the energy problem is basically shown in this slide here. What the focus of the slide is this vicious cycle between the low brain energy status, neuropathology, deterioration in cell function, cognitive decline, lower brain uptake, and we go in a vicious cycle. But this problem, as I said, is generally attributed to neuropathology getting started first, then the cognitive decline and low brain energy status. And what I would hope this slide will demonstrate to you is that the problem with brain energy intake is actually present in those people at risk of Alzheimer's disease long before the onset of the cognitive issues. So presenilin 1 carriers, for instance, even in their 30s, have lower brain glucose uptake. ApoE4 carriers in their 30s, women with insulin resistance, probably men as well, but we've identified this in women as young as 25 years old. People with a family history of Alzheimer's disease, and indeed in the healthy elderly, all of them have declining brain glucose uptake before the onset of the cognitive decline. So this precedes the issues associated with cognitive decline, and we think is contributing to it. So the question we asked is, what about ketone uptake? Because ketones are the brain's alternative fuel to glucose. Just by way of refresher, ketones are derived from dietary or stored fatty acids. In the case of the project that I'm going to describe to you, we use specifically a medium chain triglyceride that is ketogenic. Not all medium chain triglycerides are ketogenic. The ones we used contain C8 and C10 fatty acids, which produce acetoacetate, which is in equilibrium in the blood with D-beta hydroxybutyrate. One can purchase now on the internet salts of different ketones and esters of ketones that are usually in the beta hydroxybutyrate form and will equilibrate with acetoacetate. And these will in fact produce some acetone as well. The role I want to talk to you about today is ketones as fuels, but they are also lipid substrates, they're signaling molecules, and they have epigenetic effects. The method that we developed to assess ketone metabolism in the brain involves using a ketone PET tracer, acetoacetate labeled with C11, carbon 11. This is a short-lived isotope, so we do this injection first. We do the scan over 30 minutes with blood sampling. There's a washout period so that all the residual radioactivity is removed from the body over this period of time. And then the FDG PET is done afterwards. So this is a 2-hour sequence, which the person is in the same physiological state in the beginning as at the end. So these are directly comparable measurements. And usually within a day or two, often on the same day, we do the anatomical, functional MRI and diffusion MRI. So by way of summary of the data that we have on glucose uptake by the Alzheimer brain versus ketone uptake, CMR is the cerebral metabolic rate, GLU of glucose, ACAC is acetoacetate. This is the value. The values are shown here for the uptake of glucose and ketones before giving a ketogenic MCT drink, a single drink, then after the mixed C8 and C10 or C8 alone. And you can see in this circle that instead of having the yellow to orange color for glucose uptake, the parietal lobe in these individuals is in more like a green color, showing the deficit in glucose uptake. And it doesn't change when you give the KMCT. Whereas when you look at the ketone uptake, the values are lower throughout the brain because ketones are only contributing about 3 to 5% of brain energy requirements. So these values are totally normal that they would be lower. But you can see that the ketones are getting into the brain. So this area, particularly in the parietal lobe, which was defective for glucose uptake, there's no problem getting ketones into the brain with either of these ketogenic MCT substrates. So the cells in this area that were struggling to metabolize glucose have no problem metabolizing ketones. So schematically, the way we visualize this issue is that the brain should be like a hybrid car. It should be functioning on 2 different fuels. But in Alzheimer's disease, the metabolic switch is defective. So it should be functioning about 70 to 95% on glucose. But the problem exists starting at the beginning of Alzheimer's disease. With ketones, it should normally be about 5%. If you do intermittent fasting or on a low carbohydrate intake, then the ketone contribution will be much higher. But there's no decrease in brain ketone uptake. And this has been demonstrated not only in the PET studies that I've just shown you, but also in work that I've just reviewed here and don't have time to discuss with arteriovenous difference and gene expression studies as well. So 3 different methods confirm that ketone uptake by the brain, the capacity to take them up, is normal in Alzheimer's disease. So this ability to switch from glucose to ketones is the challenge that we're trying to overcome. By way of summary, the study that we did was called the Benefic study, and what we showed with this randomized controlled trial is that the ketogenic MCT increased blood and brain ketones. This randomized controlled trial used 30 grams per day of ketogenic MCT or a matching placebo. We fed it for 6 months. We had excellent adherence. So 79% of people completed the trial in both arms. Excellent safety profile, cardiometabolic safety, that is. And we showed that we could reduce the brain energy deficit, the brain glucose deficit, by about 40%. The MCI were heterogeneous, so not all had an amnestic problem, but some had other cognitive issues. I'm not going to show you the details of the demographics, but they were well matched. This graph is showing that 6 months after the start of the study, the ability to produce ketones in response to a dose of ketogenic MCT was absolutely intact, something that was not known at the time. So the body can produce ketones from ketogenic MCT without difficulty under these circumstances. And this is increasing the ketone uptake by about twofold, which corrects the glucose deficit in the brain by about 40%. The cognitive outcomes are obviously of interest in this situation, and what we're showing here is that 5 out of 5 of the cognitive domains were improved. Here I'm showing 3, and we'll show on the following slides the 2 other cognitive domains as well: processing speed and attention. So episodic memory, which is assessed with the free and cued recall test, improved by 1 word on the ketogenic MCT group. This is the change in words from pre to post test. So 0 was the value for the placebo, no change, a 1-word improvement out of 16 on the ketogenic MCT. Verbal fluency actually went down on the placebo group and improved on the ketogenic MCT group by a difference of 3 words. And the Boston naming test showed no change on placebo and a 1.3 word increase on the ketogenic MCT. This is with 43 people in the placebo group and 39 on the active group. I point out that this change in free and cued recall has been reported by people, other people besides us, that an improvement of this sort should be associated with a delay in the onset of Alzheimer's disease by 1 to 3 years. So this is definitely something to be followed up in a longer term study. Critically, for understanding what ketones are doing for cognition is the relationship between the change in cognition shown on the y-axis and the change in ketones shown on the x-axis. So these data are for plasma ketones, which almost any laboratory can measure. I also have the data for the brain, but won't be showing them. But that's more obviously not available to everyone. So I thought what was most useful would be to show the change in plasma ketones because they're tightly correlated with brain levels of ketones as well. So the ketones increase in the ketogenic MCT group. They don't change on average in the placebo group. And there's a positive straight line relationship with the improvement in episodic memory, executive function, and language. Clearly, these are not outstanding correlations, but they are present, and this is a pre-post difference, so it's actually fairly impressive to see. And there's also some people on the ketogenic MCT did not show a significant improvement in the ketone levels in the plasma. So this level of ketones produced by 30 grams per day is having some benefit in some people, but perhaps it will be possible to improve on the ketogenic efficacy of this type of treatment in future. Turning to the other 2 cognitive domains, what we see for one with processing speed is that this is a map of the tracts, the white matter tracts in the brain, and the uptake of ketones in them. So this has never been shown before, but our imaging colleagues have been able to actually measure the ketone uptake and the fluorodeoxyglucose uptake in these tracts. It's a modest level in this blue-green color in the tracts before giving the ketogenic MCT. And afterwards, you can see that they light up and the color goes up to yellow to orange, especially where the tracts enter the gray matter. So this is demonstrating that the white matter tracts are biologically active, energetically active, and can consume ketones in this situation. And when we look at the relationship between the uptake of ketones in these various tracts and the change in processing speed, we can see that these bars are showing the correlation coefficient between these two parameters, the tract ketone uptake versus the processing speed, the speed at which people process information. And in the overall white matter, we see a correlation of better than 0.5, and in some of these tracts, it's upwards of 0.6, with the important exception of the posterior cingulate, which is clearly an important area in the brain for the progression of cognitive issues towards Alzheimer's disease, in which we don't see such a strong correlation at all. So these tracts are mapped in this glass brain, and these are areas that are all affected in Alzheimer's disease, and we're seeing essentially an improvement in myelin integrity in these tracts by the presence of ketones, and it's directly related to the improvement in processing speed. And using functional MRI, we've been able to demonstrate that in one of the networks, we assessed 8 networks, including the default mode network, and in only one of them was there a significant relationship between functional connectivity and brain ketone uptake. And that was in the dorsal attention network, which is mapped here to this area in red. Here superimposed on the ketone uptake in the brain. Again, this is the first time that anyone, as far as we know, has attempted to relate the functional connectivity in a particular network to the energy metabolism in that network. And we're seeing here a strong correlation between the functional connectivity in the dorsal attention network and the uptake of ketones in that same network. And when we look at, again, the fifth dimension of cognition, the attention and processing speed, this is the Z-score for tests that involve attention and processing speed, and more specifically, the TMT, or the trail-making test, both the visual scanning speed and the number sequencing. And in all 3 cases, we see a strong positive correlation with the Z-score, or a strong negative correlation with the processing speed. So time is going down. That's an improvement in the trail-making test in relation to the functional connectivity. So there's a relationship between the functional connectivity, the improvement in cognition in relation to attention, and the availability of ketones, or the improved energy status of the brain. So if we come back to the schematic I was showing you earlier about this vicious cycle, just to remind you that the chronic brain energy deficit seems to be fed by a pre-symptomatic glucose problem. We don't know yet the relationship with the brain pathology, but I'll show you what we have learned recently in the next slide. And this is the vicious cycle that we envisage that we're trying to break up. In terms of neuropathology, we've recently had the Zetterberg lab, which is one of the key reference labs in the world, measure the plasma biomarkers of Alzheimer's disease neuropathology in this study, the Benefic trial. And as you can see quickly, there's no change in the phospho-tau 231, the phospho-tau 181, the beta amyloid 40 or 42, the glial fibrillary acidic protein, or the neurofilament light. These are the main markers that can be measured in blood. There's usually a correlation between the presence of these markers and the progression of the disease, and we're seeing that there is no change in the neuropathology by this indirect measure after having given a ketogenic MCT or the placebo in mild cognitive impairment. So in this schematic, what we're showing with the ketone treatment is that we're improving brain energy status. So we're helping to reduce the impact of brain glucose hypometabolism on the brain. We're definitely improving functional connectivity and probably white matter integrity in the brain. We don't know whether there's actually been any change in the neuropathology. By the indirect measure of plasma, that doesn't seem to be the case. And by this approach, we're reducing the impact on the cognitive domain, so improved cognitive function, and we seem to be reducing, therefore, the impact on deteriorating brain energy metabolism. We don't know yet whether we've actually delayed the progression of the disease. It's going to take a longer-term trial, but it looks like we're inhibiting this vicious cycle. So what is the context then of nutritional ketosis as a lifestyle intervention? I mentioned that we've used ketogenic MCT. You can also use salts or esters as different forms of supplements. They haven't been shown yet to improve cognition in any type of long-term study in the elderly, but this is going to come. It's going to take a placebo treatment, which at the moment I'm not aware of any placebos for these types of supplements. I think the ketone supplements are a contribution. I think we can also improve some of the issues associated with metabolic dysregulation by some carbohydrate restriction. I'm going to show you on the next slide that this is an approach that I think can be beneficial. It's also complementary with exercise, which is improving brain-derived neurogrowth factor and is also improving ketone uptake. So these are complementary, and there may be other non-pharmacological or pharmacological interventions of which ketones can contribute to improving brain energy status and hopefully slowing down the progression of this disease. So by way of a final data slide and very preliminary data at this point, what we're mapping here over 24 hours is blood glucose with continuous glucose monitoring, and the intervention was a 50% reduction in dietary carbohydrate, which improved postprandial glucose in nursing home residents. So this is a captive population that is clearly vulnerable to cognitive issues, is presumably consuming a higher glucose than they should be, and this is a one-week intervention just initially to try and assess feasibility. So we didn't change the breakfast glucose load. We did reduce glucose and carbohydrate rather at lunch, and we reduced it at supper. We didn't change the area under the curve of postprandial glucose at lunch with this reduction in carbohydrate, but we did reduce the postprandial glucose with the reduced carbohydrate at supper. So, and we also reduced the excursions of glucose all during the night. So this is just a one-week intervention. It obviously needs to be done over a longer period of time and in more than 4 people, but it is possible to reduce glucose excursions and improve glucose tolerance in nursing home residents, and we're going to try to see whether we can have an impact on cognition over in the future. So by way of summary, what we're talking about here is brain energy rescue with ketones. We think it's a key important component of cognitive health during aging. I hope I've convinced you that brain glucose deficit is a specific problem related to glucose and is not a global problem of energy metabolism, and that it contributes to the risk of Alzheimer's disease and a vicious cycle. And that there's normal brain glucose uptake has been demonstrated independently by 3 different methods, which I haven't had the time to mention, but this is now a robust observation. Ketones can improve cognition in mild cognitive impairment and in Alzheimer's disease. There are multiple studies on this now, but most of them are short-term and will need to be extended. There may be an indication of an improvement in neuropathology that has been reported 2 years ago by CSF levels of markers. So this needs to be followed up and compared with the plasma values, which we don't see any change. I think reducing insulin resistance is contributing to problems with brain glucose uptake. It's a problem that is independent of age, and it is a problem that we absolutely need to address if we want to improve cognitive health during aging. So ketones are potentially part of the solution, but we need to improve glucose tolerance and perhaps have other interventions as well. Ultimately, larger, longer-term RCTs are definitely going to be needed. So thank you very much. And I hope to contribute to the question and answer session at the end of our session. Thank you and have a good rest of the meeting. Bye now.